Okay, and now we're moving to our final event of the day, our keynote speech, which I've been looking forward to all day as well. And I'm going to pass over now to Thomas Conlon from the Michelson Prize and Grants to introduce our keynote speaker.
Thank you. It is really a pleasure to finally introduce Ricky Lewis as our keynote speaker and actually finally meet her. I believe she was as excited to accept our invitation as we were to offer it, as she had never thought of some of the molecular technologies that we've brought up today as a tool for contraception. And she is quite the cat person herself, as you'll find out during her talk. We've both spent a career in genetics that has overlapped in the gene therapy, gene transfer space, and it turns out it's overlapped in other ways, as we just figured out that one of her students ended up being one of my teachers in graduate school. So 3 generations there. And she's really played an important role as, I would call it as a translator from the researchers to the general public for the things that we try to do in genetic and molecular medicine. That has really helped people understand the concepts, the technology, a little bit of the science behind it, but mainly the goal and the successes that we've had. Having received her PhD and master's in zoology, PhD in genetics from Indiana University, she's led an amazing career, eclectic career, including a science writers for the PLOS library and the Genetic Literacy Project, which I've mentioned to a couple folks here already, that I really recommend signing up for their daily digest if you want to learn more about how genetics is being used in medicine, agriculture, around the world. It's a great way to learn about all those activities that are ongoing. She also writes for the Medscape Medical News as well. In addition, she's contributed to a number of textbooks. She's beginning to write the 13th, the 13th edition of her genetics textbook, and teaches genetics online, the ethics of genetics, and sees the occasional genetic counseling patient as well. So is highly involved in the space, and it's really a pleasure to introduce Ricky Lewis.
Well, thank you for inviting me to come here to show you pictures of my cats. My husband and I take in FIV cats because there's so much misinformation out there, and we're pretty active in explaining that they can be healthy for many years. So these are 3 of them, Juice, Artie, and Romeo. Artie and Romeo are getting older, and they're absolutely fine. Juice, unfortunately, passed away. So when Joyce asked me to give this talk, as Thomas just said, I initially said no because I didn't know what anything I did had to do with pet contraception. And she said, well, what about gene therapy as sort of a one-time fix? And I said no again, and then I couldn't get it out of my head. I kept thinking about it, and I went to the Michelson page, I read all the abstracts, and I thought, this never dawned on me, but it makes a lot of sense. And then when I thought about it more, I realized that it's a fantastic story that's now coming full circle, because we wouldn't have gene therapy in people if it wasn't for dogs. So that's where my talk begins. My book, The Forever Fix, is mostly about a young man named Corey Haas who recently graduated from high school. He was treated for with gene therapy for a form of retinal blindness when he was 8 years old. When Corey was born in 2000, this Briard sheepdog on the left, Lancelot, was treated with the gene therapy. He was the preclinical model. The researchers at the University of Pennsylvania would treat one eye at a time, obviously, so if it went wrong, they didn't blind the animal. And these dogs that I'm talking about are models, but they have natural mutations. They're not made to have a genetic disease. They are born with them. Lancelot was a very, very sad puppy because he was completely blind, and he would just sit in the corner and he wouldn't interact with anybody. And if you have my book, The Forever Fix, my favorite chapter is 16, called Christina's Dogs, and it's about the development of this canine model. So Lancelot was the first one treated, and this is just a great story. The researchers developed all these biometric tools to figure out if the dog would be able to see anything. And one day the technician came running out screaming, "The dogs are spinning, the dogs are spinning." And what happened with the pups is that when they woke up, and all of a sudden, after being blind for months and months, they had half of a visual field. So to see the entire visual field, they started to spin. So they got rid of some of the tests that they'd planned, and they just looked at the behavior of the animals, and they can tell right away that they'd got their vision back. So it worked really, really well in dogs, and Lancelot founded a dynasty. The dog to the lower right is named Mercury. I think he's like a second cousin, twice removed or something. There's a lot of these dogs. And the researchers adopt them as pets, so they have a pretty good life. Mercury was at a retinal blindness meeting I went to, and I think it was Corey or some other child who'd been cured, was running around in front of the room just like this, and Mercury was running after him. And the woman who opened the conference said, "That's no longer the blind leading the blind, it's the sighted leading the sighted." Because Corey would never have gotten his sight back if it wasn't for these wonderful dogs. So that's one of my favorite dog stories. This one is even better. This is an amazing story. And if you just Google that disease with my name, you'll find the article I wrote on it about a month or two ago. It's a horrible disease called myotubular myopathy. And 3/4 of the little boys born with this don't survive beyond a year because they can't use their muscles, they can't breathe, and they suffocate. And it's just absolutely horrible. So this boy named Joshua Fraze was born with this disease, and his parents were sort of well connected, and that helps when you're a fundraiser. Allison was a publicist for Guns N' Roses, and Paul Fraze was in the NFL. He was in the Jets. So they had a lot of connections. They were able to do a lot of fundraising, but they actually have developed a gene therapy because Allison's mother was watching CNN one morning, and she saw a report on the artificial bladder, which was developed here in Boston by a regenerative medicine guy named Anthony Atala. And she just, I think there wasn't email yet when this happened, so she must have contacted him some way. He became interested. He put together a scientific team that were interested in this particular disease, and by word of mouth, one of the researchers on the team was at a conference, gave a talk about this disease, which is distinctive because these skeletal muscle cells, the nucleus, instead of being smashed to the side, the nucleus is in the center. There's central something or other myopathies. So this pathologist showed a slide of muscle from a boy, and there was a veterinarian in the audience who said, "Wait a minute, in Saskatchewan, there's a bunch of Labrador retrievers with sick puppy syndrome." That's what it was called there. So connections were made. Allison somehow found this dog named Nibs, to the left there, Chocolate Lab, Nibs, female, living on a farm in Saskatoon, and she went up there to meet the owners, a farmer and his wife, and the dog, and when she explained the story and how her son was dying and nobody was working on his disease, they gave her Nibs. So she brought Nibs back to Wake Forest. Nibs lived on a farm, and they bred a dog colony from Nibs, and they developed the gene therapy, they tested it, and it worked so well that they've got a male dog named Ricky, like me. Male dog named Ricky was cured because of the gene therapy. So because he was so vigorous, he was able to have puppies. So they were able to get a dog colony with homozygous females, and it sped up the gene therapy. The gene therapy, it was published on the first 2 cases in September, and I don't know why the media didn't pick up on this. But these little kids who would die by age 1 are now completely normal. They're 3 years old now, I think, and their respiratory tests, everything is just working because of the efforts of this family and the role of the dogs. And Jonathan lived to the age of 16, which was pretty amazing. But this one family and the dogs did it all. So vectored contraception goes beyond pets, as I'm sure you guys out there know way more about this than I do. But the upper left is my Panda. Panda died of FIV a year ago. But it's also being done for farm animals, wild animals, and zoo animals. I'm a big hippo freak, so they end up in all of my talks. when I give talks, I never know the level of the people in the audience, so I apologize if I'm talking down because I know you know this. But I found if I give a lecture, like at a library or something, and I don't mention what RNA is, nobody understands anything. So the central dogma, joke there, dogma, of molecular biology is that DNA encodes RNA and that encodes protein. You have to know that. And the base pairing rules, A with T, G with C, are absolutely critical to any biotechnology. So those are the two things anyone has to know in order to understand any of the gene therapy. And the protein, of course, is what gives you the phenotype, the trait, or the disease. Okay. So there's different types of targets in the vectored contraception. Because we're working with genes, you're looking at proteins or the protein portion of a glycoprotein or glycolipid. If you're talking about steroid hormones, they're not protein or peptide hormones, but you can alter the enzymes that are used in their biosynthetic pathways. There are molecules to help the sperm mature and swim, and there are molecules that help the sperm bind to the egg, and then there's the interaction between the egg and the sperm and whether the fertilized egg can implant, and all these different places where you can intervene. So this is the hypothalamic pituitary gonadal axis, which is in every textbook I think I've ever written, but I'm sure, again, you guys know more than I do. But I put the green up here to show the various points where some sort of a gene therapy or other type of gene silencing therapy might be used to intervene in the reproductive process. So you've got the hypothalamus, and it's the releasing hormone to the pituitary hormones, affects the gonads, and then you go down through the gametes and the fertilization. I'll get back to that. So these are the familiar targets, the glycoprotein layer that's around the egg, the receptors, and this is somewhat species-specific. So why target the DNA? Well, a conventional vaccine, it doesn't work that well on everybody and may require several boosters. RNA molecules themselves break down fairly easily with nucleases in the cell, so it might not be a lasting effect. But DNA, a change will persist as the cell divides if the DNA is integrated into the chromosome. Okay. So there's various methods, and it's getting more confusing by the day. Traditional gene therapy, such as before 2012 when CRISPR was invented, meant introducing a gene with usually through a viral vector, and the gene gets into the cell, doesn't necessarily integrate into the chromosome. It can stay outside the chromosome in a circle called an episome, and the cell either doesn't divide or it would divide and perpetuate the gene, but you would measure it to see if there was some kind of an effect. There are a whole bunch of technologies that silence genes, and actually one of them has led to a treatment for Duchenne muscular dystrophy that's somewhat controversial about how much it actually helps, but I think the approach is valid, and it really just has to be optimized for it to work on various diseases. Gene editing, which you've heard a lot about, is the CRISPR-Cas9 system, is more powerful than traditional gene therapy because in traditional gene therapy adds a gene. CRISPR enables you to add a gene, replace a gene, or disable a gene. So it's very specific. It lands at a particular point in a genome, and it does whatever the researcher wants it to do, hopefully without messing up the rest of the chromosome. So those are the three basic methods. Unfortunately, there aren't very many good illustrations of gene therapy. I know because I've looked for all of my articles, and this is just about the best one that shows a factor 8 and a clotting factor. And the gene that you want, you just put it inside. It's not that easy, but you put it inside the virus. The virus takes it into the cell, integrates into the nucleus, and the cell will make the proteins that you need to have the effect that's desired. So this slide just shows the distinction between the classical gene therapy and the gene editing that can do other things. The first gene therapy trial was in 1990 for adenosine deaminase deficiency. Most people, I don't think, realize it's been around that long. The first FDA approval of a gene therapy for a genetic disease was in December 2017. So it took 27 years. So people, I mean, if you saw the 60 Minutes episode about CRISPR, you'd think you could go to CVS and Walgreens and get a CRISPR treatment right off the shelf. It's a long, long way out. It's been 4 years, guys. I mean, the other thing took 27 years. So we're not going to be seeing the CRISPR treatments anytime soon, but there is talk that they may make traditional gene therapy somewhat obsolete, but I don't think that's going to happen. Okay. The delivery methods, I think you heard at some of the talks today, and you'll probably hear tomorrow. There are basically 4 types of viruses that have been used, and some of them have run into some highly publicized problems. The adenovirus as a vector, it's a pretty big virus, led to the death of Jesse Gelsinger, an 18-year-old in 1999, and that pretty much ground the field to a halt. For about 3 years, there were a lot of problems. Jesse had a fever the night before. There's questions as to whether he should have been treated. Adenovirus is still used in some gene therapy trials, but not very many. And 2 years after Jesse died, a gene therapy trial with a retrovirus, the retrovirus landed in an oncogene and caused leukemia in three or four boys in this trial. The gene therapy worked. It cured their immune deficiency, but since children died in the trial, that stopped things as well. And the other vector just has a PR problem. Lentivirus is also known as HIV, and try convincing the public to use a disabled HIV as a gene therapy. So mostly you hear about adeno-associated virus, which is, many people think these are superior viruses for many reasons, the most compelling of which is that the outside, the capsid, can be changed in lots of different ways, so you can make lots of different types of adeno-associated viruses, and their tropisms are to all different parts of the body, and many of them go to the brain. I think it's AAV8 and 9 actually cross the blood-brain barrier, so they've been pretty eclectic vectors. So a viral vector needs to be big enough, it needs to either integrate into the chromosome or stay as an episome, and it needs to home to the place you need it to. I never gave this talk before, so I have no idea how long it takes. That's why I keep looking at my watch. I have a standard Forever Fix talk that I could give in my sleep, but this was highly specialized. So adeno-associated virus is pretty much the leader in the field. I don't need to go over the details on the right, but it's pretty clever how the DNA sequence, when you have inverted repeats, it's like a second layer of language that serves as like directions. It's almost like having a GPS, which will get me home tomorrow. It's almost like having that as the vector to get to the right place in the genome. So these are some of the tropisms on the left, and the fact that they go to the brain is huge because there's so many horrible neurodegenerative diseases, some of which I'm about to tell you about, if I remember. Oh, this first is one of the kids who was cured of his blindness. He was on America's Got Talent, and it was pretty amazing. But I saw something even more amazing. I saw him 3 years before this, 2 weeks after he had gene therapy, and it was at a fundraiser in Philadelphia for another form of blindness. And he came on and sang Don't Stop Believing, and nobody in the audience knew he had been blind. And I was standing next to Jean Bennett at the show, and she's the researcher her husband's the ophthalmologist who does the surgery, and she pretty much invented the whole thing. And so he's a very early case of a huge gene therapy success. He's an amazing kid. And here's some of the other cases that I write about very, very frequently. Hannah Sames lives 10 minutes from where I live, near Schenectady, New York. And she has giant axonal neuropathy. Like that little boy Joshua, she would probably not live beyond age 18. But she had gene therapy using, I think they used AAV2 on her. She had it exactly 2 years ago. And she's doing really well, but with these kids who've had gene therapy, it's very hard to tell because it's not, even if an experiment is controlled, the parents see the kid treated, the kid knows she's treated, they want to see all the positive effects that they can. So yes, they've seen Hannah improving. She could stand, she can walk a little, but she's doing PT 8 hours a day, and maybe that has something to do with it. And also, she's developed other symptoms since the gene therapy, because the gene therapy alters the course, the natural history of the disease, and you just don't know what's going to happen. So she's better in some ways, but she's declining in other ways. And they've treated about 8 kids now, and they're really, really closed-mouthed. They have to be about how these kids are doing. So that's little Hannah. She loves dogs. She has about 3 dogs. Eliza O'Neill was also treated in July 2016. Her gene therapy was an IV infusion into her hand. I mean, how much simpler can you get? That's just amazing. And her dad has told me that she's doing better, but he doesn't want me to write about it because every time I write about it, he gets in trouble with the funding organization in the hospital. But she was nonverbal when she was treated, and he put a post on Facebook and had to take it off immediately, but I saw it. And he was singing This Old Man with her, and she was echoing him. She was able to follow the song, echo the part that not an echo, I mean, she was answering him. He'd sing the first line, she'd sing the second, which shows cognition and it shows behaviors that she hadn't shown before. So Eliza has Sanfilippo syndrome type, I forgot what type, I think it's A, and these kids are absolutely doomed, and she is doing better since she's had the gene therapy. I don't think there were any dogs involved in that one. I don't remember the exact model. So vectored contraception has a different goal than gene therapy. You're not adding a gene, you're trying to take away a gene's function. So the goal is kind of opposite in a way. And here are some cats I wanted to mention. The one on the upper left is Butterball. She lives with my daughter. The middle one is Marbles, and she died, but she didn't have FIV. And the lower right is Trouser. Just no HIPAA laws, so I could do that. So this sort of a summary slide, I added the points in green at which you might intervene and how you might do so. The upper right, AAV, introduces instructions to male anti-gonadal, I don't remember the name, whatever, GnRH, it's the releasing hormone, gonadotropic releasing hormone, antibodies, and it's intramuscular injections, and it leads to atrophied gonads. The anti-malarian protein is a shot in the middle, and that blocks the maturation of the ovarian follicles. And I think most interesting, the anti-zona pellucida one, is instead of, well, monoclonal antibodies, if your body has an antibody response to an antigen, it's polyclonal. You make different antibodies to different parts of the antigen. Monoclonal antibody technology takes a single type of antibody and mass produces it, but it's complicated. You have to create a hybridoma cell, that's a B cell mixed with a cancer cell, and the actual giving of the monoclonal antibody is a protein infusion, because it's a protein, every week or every 2 weeks. It's enormously expensive. You have to keep doing it. If instead you could knit the instructions for the monoclonal antibody and deliver it with a viral vector, the animal will make the monoclonal antibodies on its own, and it should bind its target, disrupt reproduction, and you should be able to get a contraceptive effect from doing that. So you can see there's all sorts of places that can be interrupted with a vectored approach. Just to mention what CRISPR is, it's a borrowed bacterial defense against bacteria that uses short RNAs to target specific genes. The CRISPRs are actually repeats all over the genomes, and you fashion the guide RNA to take the scissor, which is the nuclease, to a certain part of the genome and cut things out. So problems with it, there was a big paper actually last week showing effects downstream from where you want to cut. So there's a lot of bugs that have to be worked out of it. But CRISPR's coming along, and behind it are 2 other techniques you don't see written about very much, talons and zinc finger nucleases. And the zinc fingers have actually been around since 2009, and there are companies, there are clinical trials going on, so we'll be seeing these types of technologies coming along as well as the gene therapy. The lower right is Trouser in a lasagna box. It's in all of my talks. Okay. And here's a slide of what the gene editing kind of looks like. I think it's kind of a bad acronym, because I never remember what any of it stands for, and I don't think many other people do either. It's very descriptive, but it's hard to remember. So gene silencing, that wasn't the topic of my talk, but it just seems to me such a promising approach, and there's so many ways of going about it that something's got to work eventually. And gene silencing, I think, is what you want when you're dampening gene expression rather than, revving it up or replacing something. Antisense RNAs bind to and block specific mRNAs, so it's countering gene expression. And then there are synthetic RNAs, with the fantastic name Morpholino. I don't even know what that means. I thought it was an Italian restaurant when I first read about it. But it's a short RNA with this sort of the sugar phosphate backbone. Is not exactly like a nucleic acid. It's a synthetic thing. But it works. It fools the cell, and it will shut certain functions off. MicroRNAs are fascinating. They are natural. They're in the body. They're 21 to 22 nucleotides long, and they clamp down on sets of mRNAs, and in that way they can control gene expression. So these things are enormously complicated because they can affect more than one gene at a time. And then we have the monoclonal antibodies, which, when they bind certain proteins, can effectively silence them. Okay. And I think, that was the last slide. So this is just a picture of all the places that I write for. And I'm happy to take questions.
Just sort of fascinating. Thank you so much. It's so exciting. If you haven't read Forever Fix, I strongly recommend it. It's packed full of fantastic information, but it's also a really good read. It reads like a novel. It's a page turner. It's beautifully written. So
thank you.
I very much recommend it. Okay. Does anyone have any questions for Ricky? Aha, we have one right here.
Thanks for a fabulous talk. Someone brought this up earlier today when we were talking about gene therapy for contraception in animals, is that the public perception of modifying genes or working with genes terrifies people. There's a lot of misinformation about GMOs. There's a lot of angst about we shouldn't be fooling around with this kind of stuff. One of the challenges we have in developing these projects is to be able to explain them in a way that I think you've done very well in your book that really gives a perspective on the risk-benefit of this type of work. Could you talk about how you see the evolution of public opinion on gene therapy over the last, say, 10 years as some of these fabulous forever fixes have become publicly aware? People have become aware of them.
That's a great question. The past 10 years, people don't know what gene therapy is. I know because my book hasn't sold well. It's only sold to people who already know what gene therapy is. So, I mean, that's how I know. The average person has no idea. But in the past year, so much has happened in the past year. And not just CRISPR, two things. One is the direct-to-consumer genetic testing, which I initially hated, but I turned around. So many people are having their DNA tested now that it's forcing them to learn. And if you look at some of those websites, like 23andMe, some of them are really good, really well done. So people are teaching themselves. The second thing is the Golden State Killer, because we're catching horrible criminals based on stored DNA. So even though there's an outcry from the lawyers and the bioethicists and the politicians about the privacy, good is coming of testing DNA. And if we could just keep stressing the good, I think the public's going to come around. We'll have more gene therapies. There were 2 this past year. The other one was the chimeric antigen receptor technology to treating cancers and the blindness. There's going to be more. There are already some in Europe. There's the ADA deficiency, the different types of SCID. I mean, they're coming, the myotubular. They're going to start coming, I would say, 2 or 3 a year. So there's going to be some critical point at which people are going to say, hey, we could do good with manipulating genes and forget about the corn and all that stuff there. So I think there's going to be a change, but I'm overly optimistic about genetics. So there is a lot of fear still. My professor in graduate school called it the triple-headed purple monster mindset. It's just DNA causes monsters to form, and that people don't go beyond that. But when they're doing their own testing, meeting their relatives, whatever, I think it's changing. People are doing it themselves and seeing real people, real tests, rather than just boring textbooks. So that's a good question. Yeah.
Okay. Another question down the back.
A fascinating talk. You showed the example of where a animal disease helped a human. Mm-hmm. Is there any type of national database or where scientists are putting together these very unique but rare diseases where animal models may discover something or human model, disease prevention through genetics analysis could actually show this kind of very unique One Health bond between animals and humans and help promote gene therapy?
I don't know if there's actually a database for that. There must be. I'm thinking of the National Organization of Rare Diseases, NORD, is a great organization and a great website. And at the bottom of their page, you get like an alphabet, and you can click on whatever disease you want to look up, and that will take you to different animal models. Yeah, it's hard to find which ones. A lot of times when I'm writing about a disease, I'll get a news release about a rare disease and I'll want to write about it. I go to Facebook and try to get the pages of the patients. And then you get they're private groups, you have to get admitted. And then once you get on a disease page, then you can find stuff out. And sometimes you find out that there are animals. But that's a good question. I'm going to have to look that up. At midnight I'll remember that there's an organization that I'm not remembering now. There must be. One of my favorite sources is clinicaltrials.gov, and you can look up all the trials there, but that's clinical. The animals would be the preclinical. So I'm not sure.
Okay, we've got one more question here. Thank you, Michelle.
Hi. So I'm curious if this would ever be used for contraception in humans.
Ooh, good question. I think that might have a PR problem with reversibility. Like it might be a form of permanent sterilization, and it would have. Sure, there is. That's true. I bet you could invent one that you could reverse. I bet that could happen. It would have to be tested just to see if there's adverse effects, but yeah, I would think so. It makes sense. That's a good question, too.
Any other questions? If not, we will let Ricky off the hook. Fabulous. Thank you.